Hi everyone. Before we actually start to talk about deep learning models applied to molecules, let us look at the question, why is it even interesting or necessary to make predictions for different kinds of molecules? After we have done that, I will give you a short overview about the topics that we will cover in this course. At the most fundamental level, life is composed of various types of molecules that interact in complicated ways to create biological structures and functions. These molecules, for example, include larger molecules such as DNA or proteins, which are often described as molecular machineries, or small molecules such as simple sugars, which can serve as energy sources. You can see a visualization of a protein and a small molecule here on the right side. The different balls that you see in these images are the atoms of which the molecules are composed. If you don't have a large biological or chemical background, don't be afraid. In future videos, I will explain to you everything that you need to know about molecules to be able to follow the content. So for now, we just need to remember that proteins are large molecules that function as machineries that fulfill different tasks. So why do we want to predict something about molecules? Understanding the function of molecules is crucial for many different scientific fields, including medicine, biology, biochemistry, which is the study of chemical processes within and related to living organisms, or environmental sciences, for example, when studying and managing pollution. One example from the field of medicine, where it is important to understand properties of molecules, could be the following. If we want to treat a disease and develop a drug for it, we often need to search for molecules that either inhibit or activate the function of a protein. Therefore, it would be beneficial to be able to know which molecules interact with a certain protein and also to know in which way they interact. For example, do they activate or inhibit the protein's function? Another example from the field of biology could be understanding the metabolism of cells. How can a cell grow and produce all the relevant molecules that are required for cell growth? To properly understand this, we need to determine the functions of many different molecules. On the other hand, molecules often play an important role in industrial settings. For example, you could be interested in finding a material which should have specific properties such as high strength or specific electrical characteristics, which depends on the molecular structure and the molecule composition. You could use prediction models to screen a large number of different molecules for a property of interest without the requirement of executing an experiment for each of those molecules. And another example could be that you are interested in generating a specific molecule starting from a different molecule. For example, you could be interested in producing a molecule named 2,4-dihydroxybutyric acid, or short DHB, starting from malate. The DHB molecule is important for the production of many pharmaceuticals, but its natural abundance is low. On the other hand, the natural abundance of malate is high. It is thus highly desirable to find proteins that can convert the malate molecule into the desired target molecule. To achieve this, you need to find proteins with specific functions. And in this case, it is actually possible to use three different proteins and two intermediate molecules to produce DHB molecules starting from malate. In this lecture, we are mostly concerned with using machine and deep learning methods to determine the function and properties of molecules. But before we do this, I want to give you a quick overview about alternatives to using machine learning for determining molecule properties and functions. For this, we will come back to our example from the previous slide, and we will quickly look at different options that we have to determine the function of a protein or for finding a protein that can catalyze a specific reaction. For example, if we think about the example from the previous slide, we might want to experimentally confirm if a particular protein binds to the small molecule malate and subsequently catalyzes its conversion into a desired product. Biochemical experiments are one important option to answer such questions. This involves multiple steps. The first step is to obtain both the purified protein and the small molecules. First, we need to get enough of the protein that we want to study. Often, the easiest way is to have a tiny living factory, like harmless bacteria or yeast cells, that can be modified to produce large amounts of the protein for us. Once they have made the protein, we need to carefully separate it from the other parts of the cell using special cleaning and filtering techniques. This process gives us just the pure protein we are interested in, so we can be sure that anything we observe in our experiment is actually caused by that specific protein and not by something else. Small molecules can often be bought from chemical suppliers. If not commercially available, they may be needed to chemically synthesized in the lab. The second step is to determine the biochemical assay. A biochemical assay refers to the procedure that is used to detect, quantify, and study the binding or catalytic activity of a biological molecule, such as a protein. This involves selecting an appropriate method to monitor the interaction or reaction, and it includes the buffer, which is a solution in which you put the protein and the small molecule. While standard assays exist, they almost always require optimization for the specific protein small molecule pair being studied to ensure reliable and reproducible results. The third step is the execution of the actual experiment, which involves precisely mixing the purified protein, the small molecules, buffer, and other required components under controlled conditions. Additionally, appropriate instrumentation has to be used to collect data that reflects the binding event or catalytic activity. The last step is the analysis of the results to judge whether the protein actually catalyzes the small molecule. The duration for this entire process is highly variable. While running a well-established assay might take days or weeks, the complete workflow, including the potentially challenging protein purification and the assay development, can easily take several weeks to many months, depending on the complexity and novelty of the system. And the process is not only time intensive but also costly. This includes expensive specialized equipment, the cost of reagents, and the synthesized molecules, as well as substantial labor costs for highly skilled researchers or technicians. An alternative for determining whether a small molecule binds to a protein or is catalyzed by a protein is using traditional computational methods, for example, molecular docking simulations. This technique offers a way to computationally predict how a specific molecule might bind to a target protein. Docking primarily focuses on predicting the binding pose, that is, how the molecule fits into the protein and estimating the strength of that binding. And it is not directly predicted whether the protein converts the small molecule into another molecule. Now, what do we need to do, or what do we need to run a docking simulation? The essential inputs are the three-dimensional structures of both molecules involved. For the protein, we need its detailed 3D shape. We can get it from experimental techniques like X-ray crystallography, or alternatively, especially if an experimental structure is not available, we can use highly accurate structure prediction models such as AlphaFold or RosettaFold, which are based on deep learning. For the small molecule, we also need its 3D structure, but this is generally much simpler to obtain, often generated from its 2D chemical structure using standard software. Once we have these structures, the docking software goes to work. You can imagine it like trying to fit a key, the small molecule, into a lock, the protein's binding site. The program systematically tries out many different possible positions and orientations for the small molecule within the protein's target site. It does this by computationally rotating, shifting, and adjusting the flexible parts of the molecule to explore how it might fit best. For each potential pose or fit that is generated, the program calculates the score. The score acts as an estimate of how favorable that particular binding arrangement is, essentially how good the fit is predicted to be. The score reflects how well the shape matches and how strong the chemical interactions are. On this slide, we will look at the advantages and disadvantages of different methods for determining properties of molecules, including machine learning methods. We have already shortly looked at experimental techniques. For example, if we would like to know if a given protein catalyzes a specific chemical reaction, which means we want to find out if a given protein can convert a starting molecule into another molecule of interest, we could experimentally measure if this protein converts the starting molecule into a target molecule. The advantage of direct experimental measurements is that the results are usually more accurate than alternative approaches. However, the disadvantages are that it can be very time-consuming, difficult, and expensive to perform such experiments. For example, if you want to measure if a protein is catalyzing a specific reaction, you will first need to reduce and purify the protein, get a concentration of your starting molecule, and then perform the experiment. Especially if you want to test your protein against many different molecules, this can take a lot of time. We have seen that an alternative to experimental methods are non-machine learning computational methods that simulate the behavior of molecules using theoretical principles, empirical data, and statistical analysis. Advantages of these methods are that you usually get your results faster at reduced cost, and the methods are usually much more accessible and easier to execute than experiments. However, disadvantages are that usually experimental results are more accurate than computational methods, or it might be infeasible to simulate very complex interactions, and you might require a large amount of high-quality experimental data to develop these tools. In these cases, it can be better to perform a single experimental measurement. And the last alternative is the one that this course is concerned with: machine learning prediction models. That means using data, usually experimental data, to train a model to predict a certain property or activity of a molecule so that we can make predictions when no experimental data is available. The advantages of this approach is that machine learning models are capable of handling highly complex data and also, for example, handling complex interactions between molecules. Machine learning models are excellent at detecting patterns in data that we might not be able to find without such models. Once machine learning models are trained, they are much quicker than experimental methods, and they can be flexibly retrained and readjusted once new data becomes available. To come back to our example from the experiments, if you want to find out if and which molecule a certain protein can catalyze, experimentally testing the protein against a large number of different molecules can be very time-consuming and costly. And also, traditional computational methods are not very useful for this task, as the interaction of a protein with other molecules is very complex, which we cannot easily simulate. Instead, we could train a machine learning model for this task. We could use lots of available experimental data for other proteins, where we know with which small molecule the proteins interact, and train a model to predict protein small molecule interactions. Using such a model would allow to predict promising candidate molecules that the protein can catalyze. This would allow us to only test a small number of promising molecules experimentally, instead of making experiments for a large number of different molecules. Now that we know that it can be really beneficial to be able to predict properties and functions of molecules, we will have a look at a few more specific prediction tasks to get a better idea of what kind of properties can be predicted using machine and deep learning models. Let's, for example, take a protein, which has a specific function. This protein will be most efficient in a specific environment, for example, at a specific temperature and at a specific pH value. If we are interested in using a protein for an industrial purpose, we want it to be as efficient and stable as possible, and we need to find the optimal conditions. We could measure experimentally how efficient the protein is at different pH values, which tells us how high the concentration of hydrogen atoms in the environment is, but doing this experimentally can be expensive and time consuming. Alternatively, we can build a prediction model that predicts the optimal pH for this protein by using information about the protein. An important part of this lecture will be to find good ways to extract relevant information about the molecule, in this case the protein, which we can use to make a prediction about a specific property. Something else that we could be interested in could be predicting the effect of small changes to the proteins, so-called mutations. For example, we could be interested in changing our protein a little bit to achieve a protein that is optimal under different conditions but still has the same function. To achieve this, we could build a prediction model that predicts the effect of a certain mutation. And of course, these are just two out of many examples for what we can predict for proteins. On the other hand, we could also be interested in predicting a property for a small molecule. When developing a new drug for treating a certain disease, this drug should have as little as possible toxic effects. How toxic a small molecule is means, in this context, the degree to which it can damage an organism. Instead of performing costly and maybe harmful experiments, we could also use a machine learning model to learn from existing data to predict how toxic a certain molecule will likely be in the human body. An important part of this process is similar as for the protein property prediction task, to extract meaningful information from the small molecule that we can use to make a prediction for the small molecule. We will later see how we can use deep learning models to achieve this. Finally, it can also be the case that we are interested in not only predicting a property of a single molecule, but we could instead want to predict how two molecules interact with each other, for example, a protein and a small molecule. Enzymes are proteins that bind to small molecules and transform these into different small molecules, the so-called products. And hence we could be interested in predicting, given a protein and a small molecule, does the protein bind to this small molecule? Another example is predicting how much a small molecule inhibits the function of a protein. When developing a drug, we are often interested in finding small molecules that inhibit the function of a certain protein. So we could be interested in predicting how much a specific molecule inhibits the function of a protein. Now that we know that it is beneficial for a vast amount of different industrial and research areas, if we can make predictions for properties and functions of molecules, we will have a look at what kind of different molecules we will be mostly cover in this course. We will focus on biomolecules, which are molecules that are present within and produced by living organisms. They are essential for life, as they serve as the building blocks of cells and organs, carry genetic information, and perform various functional roles in metabolism and regulation. However, although we will be mostly focused on biomolecules, the methods that we will cover are in principle applicable to any kind of molecule. We can broadly divide biomolecules into two categories: micromolecules and macromolecules. Micromolecules are small, simple molecules with low molecular weight. They are important as building blocks for the synthesis of macromolecules or as energy sources and signaling molecules. Examples of micromolecules include vitamins, amino acids, monosaccharides, which are small sugars, nucleotides, and fatty acids. Macromolecules, on the other hand, are large, complex molecules with high molecular weight. Examples of macromolecules include proteins, nucleic acids, such as DNA and RNA, polysaccharides, which are large sugars, and lipids. The distinction between macro and micromolecules is generally based on molecular size and weight, as well as complexity and functionality. However, there is no strict cutoff point in terms of molecular weight or size that universally applies to classify a molecule as either a macro or a micromolecule. The classification often depends on the context and specific characteristics of the molecule of interest. In the first part of this lecture, we will mostly be interested in making predictions for proteins, which are the molecular machineries of all living organisms, which means they perform a wide range of functions critical to the structure, function, and regulation of organisms. Later, we will also investigate how we can make predictions for all different kinds of small molecules. Finally, we now briefly look at all the different topics and methods that I plan to cover in this lecture that will help us to approach the prediction problems that we have just outlined. As I said before, you don't need any biological or chemical background to follow this course, because we will start from zero and I will tell you everything about the basics that you need to know. We will see that proteins are made of sequences of amino acids, and thus each protein can be represented by a sequence of letters that represent the amino acids. As you probably know, transformer networks are powerful models to process sequences and to make predictions about sequences. This is why we will make an introduction into transformer networks, and we will learn how we can apply them to process protein sequences. We will see how we can also use transformer networks to process small molecules, and we will also look at alternative methods to process small molecules such as graph neural networks and other more traditional and expert designed methods. Since we can process both proteins and small molecules with transformer networks, we will also look at multimodal transformer networks that can process both a protein sequence and a small molecule simultaneously in the same input sequence to make predictions about protein and small molecule interactions. This is similar to multimodal models that you might know from chatbots that can also process different modalities in the same input, like text and images. We will also briefly study gradient boosting decision tree models because, as we will see, these are often very suitable to process numerical molecule representations that we obtain from transformer networks. Often these decision tree based models lead to better prediction results than simple fully connected neural networks. As I said, we will use methods to produce to process the protein sequences with transformer networks. But as you have seen in all the protein images on the slides, proteins are complex 3D structures. And the 3D structure is very important for the function of a protein. And this is why we will also cover methods to process the 3D structure of a protein and not only the linear protein sequence. Depending on how much time we will have left at the end of this semester, we will also study methods that can predict how mutations in a protein affect different properties of the protein. And finally, we will cover challenges and best practices in the field of AI and biology, and we will learn about good practices to train and optimize large deep learning models that we use to process the molecules.